Hi everyone, today we turn to the open problems in aging science. Now the field has seen considerable progress, of course, but many foundational questions still remain unresolved, and this can lead to somewhat fragmented research efforts. So what we'll be examining today is a recent, systematic effort to map these very questions, effectively creating a data-driven roadmap to guide the future of biogerontology.
So here's how we'll break it down. We'll start by looking at why a roadmap for biogerontology is even necessary right now. Then we'll get into the specifics of the systematic method they used to identify these problems. From there, we'll map out the research landscape that this analysis revealed, and we'll see some pretty big differences between well-established areas and emerging gaps. And finally, we'll talk about the future directions that this research points towards.
Okay, so first, let's set the context here. The science of aging has come a long way since those early breakthroughs, things like the discovery that dietary restriction can modulate lifespan. And we've seen major advancements more recently in areas like epigenetic clocks and pharmacological interventions. But despite all that progress, the field still doesn't really have a modern, consensus-driven agenda to help guide research and prioritize what to work on next.
And that brings us to the central question that's really driving this whole research effort. What are the most important unanswered questions in aging science today? This isn't the first time this question has been asked, of course. It actually follows a historical precedent set by Bernard Streller way back in 1977. In his work, Time, Cells, and Aging, he laid out what he saw as the pivotal questions for the field. Now, almost 50 years later, this new effort is trying to do something similar, to provide an updated framework by systematically identifying the key open problems for modern biogerontology.
So how do you even begin to map out an entire scientific field like this? It's a huge task. The researchers here used what they call a hybrid approach. They started by essentially crowdsourcing questions directly from the scientific community. But they didn't just stop there. They took that input and paired it with a very rigorous data-driven analysis of the existing scientific literature. The whole goal was to create a final list that balances what scientists themselves are curious about with what the data shows is actually being studied.
And that initial collection phase for questions was quite extensive. They used an online platform and also held a dedicated workshop with 24 experts in the field. And from that, they gathered a total of 290 distinct research questions. This became the raw list, the starting point for their curation process.
From that large initial list, they then began to whittle it down. They removed any questions that were poorly formulated, and they consolidated duplicates. This process left them with a much more refined set of 204 unique open problems, and this became the core data set for their deep dive into the scientific literature.
Okay, now this next part is really the analytical heart of the study. To figure out how much attention each of those 204 problems has received, they built a three-stage natural language processing pipeline. Here's how it worked. First, they used a model called PubMed-BERT to cast a very wide net, finding any articles that were broadly similar to each question. You can think of this as a highly sophisticated semantic search. Then for the second stage, a more precise cross-encoder model scored how relevant those initial articles actually were. And finally, a natural language inference model acted as a final check. It determined whether an article's abstract truly entailed or supported the open problem statement. So only the problem article pairs that made it through all three of those rigorous filters were included in the final analysis.
So what did this powerful analytical pipeline actually find? Well, it gave them a quantitative, bird's-eye view of the entire research landscape. And the results? They showed a striking imbalance in what scientists are focusing on. Now, this could be due to a number of factors, things like funding priorities, historical precedent, or maybe just the tractability of certain research questions over others. And it's important to understand the scale of this analysis. We're talking about over 200,000 unique PubMed articles related to aging published over a 60-year period from 1963 to 2023. When you pair that huge data set with the 204 open problems, it generated more than 40 million potential problem article pairs for the NLP pipeline to sift through.
This chart really visualizes the central finding of the whole study. There is a profound disparity in research attention. If you look at the top 20 most represented open problems, they're associated with 40.3% of all the relevant articles the pipeline identified. Now compare that to the bottom 20 least represented problems. They were associated with just 0.2% of the articles, a huge difference.
Let's put that in absolute numbers, because that makes the disparity even clearer. The top 20 problems were linked to more than 69,000 articles. That's an average of almost 3,500 articles for each of those problems. In contrast, the bottom 20 problems, they were linked to only 341 articles in total. That's an average of just about 17 articles per problem. So the key takeaway here is this immense difference in representation. It shows a very heavy concentration of research effort on a very small set of questions.
Okay, so this disparity becomes even more clear when we start to look at the specific types of questions that fall into each of these categories. This analysis allows us to directly compare the well-established research priorities with what we might call the emerging or underexplored knowledge gaps. So if we look at the most heavily researched topics, you can see they're dominated by these broad fundamental questions. For example, why do we age was linked to over 10,000 articles. Other very well-represented areas include core mechanisms like somatic mutation accumulation, the evolutionary and comparative biology of long-lived species, and the translation of findings from model organisms to humans. These really align with the long-standing pillars of biogerontology research.
Now, in contrast, the underrepresented problems tend to be more specific. They often focus on novel methods or therapeutics. Take, for instance, the question about measuring changes in the homeodynamic space. That's the body's diminishing ability to maintain stability and buffer against stressors as we age. That question was linked to only a single article. Similarly, questions about how we might break through the current human lifespan ceiling or how to prioritize which interventions should go to human clinical trials, these are barely represented in the literature. This really highlights them as wide-open frontiers for future investigation.
So the culmination of all this work isn't just a static list of questions. It's meant to be a curated roadmap to help guide future research. And it's presented as a dynamic, interactive resource that the scientific community can actually use.
The final result, then, is a curated list of 100 key open problems. The authors organized these into 11 distinct themes, and as you can see from the chart, the three largest themes, aging mechanisms, molecular mechanisms, and interventions, together account for more than half of all the questions. This really underscores where the field's current priorities lie.
And this quote from the authors really highlights a key difference between this new roadmap and Bernard Strehler's original list from back in 1977. While some of the foundational topics are still there, there's now a very clear contemporary emphasis on developing and evaluating interventions. This reflects a significant evolution in the field's overall objective, moving from just characterizing the aging process to actively seeking ways to modulate it.
One of the most practical outcomes of this project is something called the Longevity Knowledge App. It's an online interactive platform where researchers can go to explore these 100 open problems and their themes. They can access metadata, find links to related questions, and, very importantly, they can propose and discuss potential solutions. The whole thing is designed to be a living resource that fosters collaboration and helps direct future research efforts. Ultimately, what this work provides are tangible avenues for future research. It doesn't just map what we already know, it quantifies what we don't know. It offers a data-driven guide to the true frontiers of aging science. And one of those frontiers is captured perfectly by this final question here. How much do positive feedback loops in chain reactions contribute to aging? With only 23 associated articles found in the literature, this is a prime example of a deeply underexplored area, one that could be fundamental to our understanding of the whole process. This roadmap, in essence, shows us exactly where we might need to start digging.